Hello. Yes, good morning to all of you. My voice is audible. Yes, yes. Yeah, good morning to all of you. How are you? And sorry, we are just be here a little bit early. Uh, Instead of our regular schedule, we just in the previous days we met on 8 p.m. or 7:30 p.m. But today we are here by 11:30 or 11:40 a.m. So thank you all for your support. And yes, we are here because I just want to take a little bit long session. So that is why instead of taking it at the night, I think we will take it in the morning. Okay. So I have the syllabus with me. Only few things are left. Rest of the syllabus is completed. That is why I think we will do it now, and then you all will be free from now onwards, specifically for your review paper, as well as you can submit the assignments also. So let me show you the syllabus. Yes, good morning to all of you. Good morning to all. We are a little bit early because I want to complete the syllabus today. That is why I take your class on the very first. After it, I will take the remaining lectures. So I am just going to show you the syllabus. So this is your syllabus. As you all know, we have done Mendel's law, types of gene action, what is quantitative, qualitative traits, multiple alleles. We have done it also. After it, regulation of gene expression, lack of brain gene. This is also we done. Ultrastructure of cell and cell organelles, chromosome number, morphology of the chromosome, karyotype and ideogram. We already covered these things. Mitosis and meiosis has been done. DNA, RNA, DNA structure, RNA structure, replication, transcription and translation has been done. This topic genetic code is not done before, so I will take this topic today. Linkage we have completed yesterday. Numerical and structural abbreviations are also completed. Mutation is completed. Crossing over is completed. So we are left with genetic code, and we are left with these topic number 4 and 5. That is CIB technique and multiple factor hypothesis and cytoplasmic inheritance. So today we will try to complete the remaining topics only, and after it I will give you the homework which you have to submit me by this morning, or by not by this morning, by tomorrow morning, or after it you have to focus on your review paper only. Getting my point? So after it you have to focus on the review paper only. So let me share the PPT of genetic code now, because we have to start with the genetic code first. So my voice is audible to all of you. Please let me know. Are you getting me properly? My voice is audible to all of you. Are you getting me properly? Yes. So thank you, Sudhirji, and good morning to all. So without wasting much time, I am starting the today's. I am starting the today's lecture, which will be on genetic code. Plus, we will cover the remaining topics which are there. Okay. So I'm just going to start it. Anyone who is not present here, they can watch it later also. And it is my request to all of you to please write the homework along with this lecture series, so that at the end we can write it in the telegram also. So we will start with genetic code now, because genetic code was left yesterday. Yeah. So good morning to all, and we are just going to start the genetic code. Sorry. So I am just going to start with the genetic code, and I will try to send all the PPTs which I have to all of you, so that you will be able to go through these all these PPTs. So I am going to start with genetic code now. Yes. So this is the remaining part of transcription and translation. We have done transcription and translation. Along with it, this is the genetic code. So after when DNA gets changes into mRNA and mRNA gets changes into a protein or a polypeptide chain, after which we have the genetic code. So all the information, the genetic information is encoded in a DNA and is later transcribed into mRNA through the process of transcription. So, however, there are few cases where DNA is absent but the information is contained in the RNA. So, proteins, from these RNA we will get the proteins. Proteins have 20 different kinds of essential amino acids. Nucleic acids have only 4 different kinds of bases. We have adenine, cytosine, guanine, uracil or thymine. So, basically the amino acids proteins consist of 20 different amino acids and nucleic acids have only 4 types of bases which are present, which are purines as well as pyrimidines. These 4 bases have been considered as the alphabet of the language of nucleic acid and 20 amino acids are the alphabets of the language of proteins. Getting my point? So these amino acids which are there for the for polypeptide chain or for proteins, these 20 amino acids will be coded here. So what is the sequence of nucleotides on the mRNA will be decided the sequence of the amino acids in a protein. So the sequence which will be there, the sequence of nucleotides, that means adenine, thymine, cytosine, guanine, the sequence of nucleotides on the mRNA will decide the sequence of amino acids in a protein chain, and this leads to the discovery of what we know as genetic code. So basically the sequence which are there in the RNA or in the mRNA, they will be coded by the amino acids in the protein chain. So the genetic code works to prepare a dictionary for translating the language of RNA into the language of protein. So language will be coded into the protein with the help of amino acids. So what is basically the genetic code? The sequence of nitrogenous bases in mRNA molecule which encloses information for the synthesis of protein molecule is known as genetic code. So in the mRNA, we have nitrogenous bases. We have adenine, cytosine, thymine, guanine. Or you can say, in the mRNA, we have adenine, cytosine, guanine and uracil instead of thymine. So, the sequence of nitrogen bases in mRNA molecule which encloses information for the synthesis of protein molecules. From mRNA, what we will synthesize? We will synthesize protein molecule with the help of these genetic codes. So, the nucleotide or nucleoside sequence in mRNA which codes for a particular amino acid is known as codon. Now, what is codon? It is the particular amino acid which codes for the sequence which is there in the mRNA. So mRNA has the sequence. We have adenine, guanine, cytosine, uracil. So these sequence will be coded with the help of a codon. So the main problem of genetic code was to determine the exact number of nucleotides in a codon which codes for one amino acid. Basically 3 nucleotides they will code for the one amino acid. So let us do it now. A single code consists of only one nucleotide for just 4 codons. These are insufficient to code 20 amino acids. If you are using only one code for a one nucleotide, then it will become insufficient to code for 20 acids, 20 amino acids which are there. Similarly, a combination of two nitrogenous bases provides only 16 codons which are still insufficient for 20 amino acids. So it was the scientist George Gambo who in 1954 pointed out that possibility of three-letter code That is, each codon, that means each amino acid, consists of 3 nitrogenous bases. Each codon consists of 3 nitrogenous bases. So this will give us 64 codons, which are more than enough to code for 20 amino acids. So we have 64 codons which will code for 20 amino acids. So this is a single code. So if we are taking it singly, then only we will get 4 codons. If we are taking it double, like adenine and adenine double, only we are getting 16 codons. But if we are taking triplet, that means we are taking triple nitrogenous bases, like this is A, adenine, guanine, uracil, uracil, guanine, uracil, adenine, adenine, uracil. If we are taking 64 codons, if we are taking triplet codons, sorry, triplet nitrogenous bases, then we will get 64 codons. These 64 codons, they will code for 20 amino acids. So we have 20 amino acids, 1, 2, like phenylalanine, leucine, isoleucine, methionine, valine, alanine, threonine, proline, serine, tyrosine, histamine, histidine, glutamine. So with the help of 64 codons, we will able to code 20 amino acids. If it will be only possible if we are taking the nitrogenous bases triplet in nature. If we are taking the nitrogenous bases duplet in nature, then we will code for only 16 codons. That means only 16 amino acids. But we have 20 amino acids. For it, we have to take the triplet nitrogenous bases to make it a codon. So the triplet code is the most acceptable one, as it has found support from an experimental evidences of both genetic and biochemical nature. The genetic code has been experimentally deciphered and perfected by Baron Nirenberg, Holly and Hargobind Khurana. Now who is Hargobind Khurana? He is an Indian scientist, and he was awarded Nobel Prize for Medicine and Physiology in 1968. So Hargobind Khurana was the one who was able to tell us about this genetic code. Along with Nirenberg and Holle, Hargobind Khurana, the Indian scientist, was able to let us know about the genetic code basically. Now, the genetic code has been cracked or deciphered by the following kinds of approaches. Like there are some theoretical approaches, in vitro codon assignment, in vivo codon. Let us discuss them also. So this is, are you getting it? Okay. Now we have to start with the theoretical approach. So there was a scientist George Gambo proposed the diamond code and triangular code and suggested an exhaustive theoretical framework to the different aspects of a genetic code. So he suggested the following properties of the genetic code. He suggested the following properties. So a triplet codon corresponding to one amino acid of the polypeptide chain. So a triplet codon, according to his observations basically, a triplet codon corresponding to one amino acid of the polypeptide chain. The direct template translation by codon, amino acid pairing, translation of the code in an overlapping manner, degeneracy of the code. What do you mean by degeneracy? That is, an amino acid is being coded by more than one codon. Like in this diagram, UAA, UUUG, they both code for leucine. So amino acid is coded by more than one codon. Similarly, here leucine is again coded by more than one codon. So this is single codon, CUU. This triplet is a single codon, but leucine is coded by more than one codon. Then we have planarity of nucleic acid and primary protein synthesis. Universality of the code, that is code being essentially the same for different organisms. Universality means the code will remain same in all organisms. Then another assignment is in vitro codon assignment. So it is divided into three parts. Number one, discovery and use of polynucleotide phosphorylase enzyme. Codon assignment by the homopolymer, codon assignment by the heteropolymer. Then assignment of the codons with no sequence. This is the discovery and use of this enzyme, which is polynucleotide phosphorylase enzyme, by the two scientists, Managu and Okoa, who isolated an enzyme from the bacteria that catalyzes the breakdown of RNA in the bacterial cells. This enzyme is called polynucleotide phosphorylase enzyme. So both they found that outside of the cell, with high concentration of ribonucleotides, the reaction could be driven in reverse and RNA could be made. So these are their observations basically. Then the in vivo codon assignment. In it also, the same code is used by amino acids. So basically this is the properties of the genetic code which you have to keep in mind. So the genetic code possesses the following important properties which have now been proved by definite experimental evidences. So all these points are correct regarding the genetic code. Number one, the genetic code is triplet in nature. Okay, so the code is triplet. It consists of 3 nitrogenous bases. The code is degenerate. The code is non-overlapping. It will not overlap with other codons. The code is comma-less. The code is non-ambiguous. The code is universal. It has some polarity. It has a start codon as well as the stop codon. The start codons will start the reaction as we have seen in the transcription and translation also, and we have some terminal code also that will stop the reaction. So the number one is the code is triplet. So a triplet or 3-letter code was first suggested by Gamow in 1954. A codon of the present day genetic code that specifies one amino acid in a polypeptide chain comprises a sequence of 3 nitrogenous bases on mRNA in a specific sequence. The first experiment evidence supporting the concept of triplet code was provided by Crick and the co-workers in 1961 in T4 bacteriophage. The code is degenerate. Now what do you mean by degenerate? When a particular amino acid is coded by more than one codon, it is called degenerate. Amino acid like leucine, valine, it is coded by more than one codon. So instead of single codon, because we have 64 codons and we have 20 amino acids, so it means amino acids will be coded by more than one codon. So it is called degenerate. Example, tryptophan and methionine, which have a single codon each, and all other 18 amino acids, they have more than one codon. Only tryptophan and methionine have single code. Otherwise, all the other amino acids, they will have more than one codon. Now, the code degeneracy is basically of two types. We have partial degeneracy as well as we have complete degeneracy. Now what happens in partial degeneracy? It occurs when first two nucleotides are identical, but only the third nucleotide is different. So CUU, CUC. If you are able to see the first two nitrogenous bases, the first two nucleotides, they are same, but the change is only in the third nucleotide. So that means they both are coding for a single, for a one amino acid, which is leucine. So they are partially degenerate or degeneracy. It means when a particular amino acid, that means leucine, is coded by more than one codon. And what is complete degeneracy? It occurs when any of the 4 bases can take third position and still code for the same amino acid. So if you are able to see here, UC is common in the first two nucleotides. The only difference is in the third nucleotide. Here we have uracil, we have cytosine, we have adenine, and we have guanine. So the third position is changed, and in third position we have four bases, we have four nitrogen bases which are present. So all these three UCU, UCC, UCA and UCG, they will code for the amino acid serine. So they are completely degeneracy. So the following table represents the occurrence of multiple codons for different amino acids and clearly illustrates the degeneracy of the genetic code. Suppose now we have the amino acid arginine, leucine and serine. So they are coded by 6. Number of codons are 6 codons. So they are coded by 6 and total number of codons will be 18. So I like this, we will get the 64 codons that will code 20 amino acids. Only methionine and tryptophan is the exception, which will be coded by only one codon. Otherwise all will be coded by more than one codon. The next property is the code is non-overlapping. Now what is non-overlapping? The code is non-overlapping, which means that the same letter is not used for different codons. The same letter will not be used for the different codons. So this is ACA. So this will code for one codon. Now it will not happen that the next codon will start from ATG. No, it will only start after when the three bases we counted for a single codon. After it we have to take the next three bases. So similarly there will be no overlapping. Okay. So first three will counted single codon and then next three will be counted for the next codon. The evidences are available to show that translation of a genetic code in an mRNA begins at the correct point and there is no overlapping of codons. That is, the genetic code is non-overlapping in nature. Also, the code is non-overlapping, but in bacteriophage, it is an exception in case of bacteriophage, there is possibility of overlapping genes and codons. After it, the next property is the code is comma-less. Now, what do you mean by comma-less? That the sequence will be in continuation. There will be no full stop, no comma in between the sequences. In between the genetic code, there will be no any type of comma or something. In other words, no codon is reserved for these punctuations. So you have to keep this in mind that the sequence is in continuity. The code is non-ambiguous. Now, what is non-ambiguous? The genetic code inside the cell medium is said to be non-ambiguous because a particular codon always codes for the same amino acid may be coded by more than one codon. But one codon never codes for two different amino acids. Yeah, please keep this thing in mind that it's okay that two codons they will code for one amino acid, but one codon will always code for, if AUG is coding the arginine, they will always code arginine in every organism. Getting my point? If CUC is coding for histidine, then it will code for histidine only. So the code is non-ambiguous. The code is universal. So same genetic code is found valid for all organisms ranging from bacteria to man. So the code, suppose AUG is coding for arginine, then it will also code for arginine in case of bacteria also. Such universality of the code was demonstrated by Marshall, Kasky and Nirenberg in 1967, who found that E. coli and guinea pig amino acyl tRNA use almost the same code. So they found this, the codes are universal in nature. The next property is the code has some polarity. So the code is always read in a fixed direction, that is in the 5' to 3' direction. In other words, the codon has a polarity. So reading from left to right and right to left will specify for different amino acids. Now some codes act as start codons, some will act as stop codons. So where the start codons are present, from where only the reaction will start. So in most organisms, AUG codon is the start or the initiation codon. The polypeptide chain starts either with methionine or formyl methionine, because AUG codes for methionine. So the reaction will start only from methionine, and after it we have some similarly GUG, it codes for valine. It codes for valine, but when normal AUG codon is lost by deletion, then GUG is the used as a initiation codon. So we have only two initiation codons. We have either AUG, which codes for methionine, but if AUG is not present, then GUG, which codes for valine, it will start the reaction. Okay. After it we have some codon acts as stop codons also. So there are some codons which will stop the reaction. So the stop codons are UAG, UAA, UGA. These are the stop codons, are also known as the termination codons, because they will stop the reaction. So they do not code for any amino acid, rather when they are present, the reaction will stop. The transcription translation will stop. After which we have some initiation and termination codon. A reading frame of the genetic message always has a specific start or stop signals for protein synthesis. The initiator and terminator codes are known as signals, and this phenomena is known as punctuation. So punctuation helps in demyelinating the different histones on a polycistronic mRNA. The signals or spacers do not occur between the codons of a message, but initiate and terminate larger sections of the message between the functional genes. So these are the AUG, UAA, UAG, UGA, they are the start and the stop codons. So AUG, which codes for methionine, is the start codon. It will start the reaction. And UA, UAG, UGA, they will stop the reaction. So since these stop codons UAA, UAG, UGA, they do not code for any amino acid, they are also known as nonsense codons. Okay, so they are known as nonsense codons also, because they are not coding for any amino acid. So signals of stop or termination codons are read by proteins called release factors. Stop signals are not read by tRNA molecules. In prokaryotes we have some factors which are known as the release factors. These factors, they will recognize the UAA, UGA, they will recognize the stop codons and the reaction will stop. Now gene mutations we already discussed yesterday also. It is an alteration in the sequence of the base in a gene due to change, removal or insertion of one or more bases in a gene, resulting in a altered gene product, also called gene mutation. So mutations are heritable permanent change in a genomic sequence. It leads to the formation of abnormal proteins or non-production of proteins. It may occur in either introns or exons or even mitochondrial genes. We have already discussed mutation yesterday, so I am not going to start it again. So we have point mutation, we have frame shift mutation. We have done these mutations yesterday also. So here there is a change in the single nucleotide only. So we have silent mutation, in which we are not able to get the result. So when the codon containing the change base may code for the same amino acid, it is known as silent mutation. This occurs because multiple codons can code for the same amino acid. Then we have missense mutations also. What happens in missense mutation? So if you are able to see here, also there is a mutation. Instead of adenine we have guanine, but still they code for glutamate. But in case of missense mutation, we will get different amino acid. So instead of glutamate, we will get asparagine. So when the codon containing the changed base may code for different amino acid, it is known as missense mutation. The example, if the serine code GAA changes to be GAC, so instead of coding for glutamine or glutamic acid, it will code for the aspartic acid. So these are known as missense mutations. Then we have some nonsense mutations also, when the codon containing the change base may become termination codon. So like here it code is for the glutamic acid, but if T is present, it will stop the reaction and we are not able to get the remaining amino acids. So if serine code UCA becomes UAA, if C is changed to A, UAA is the stop codon which leads to the termination of the protein or termination of the translation basically. Then frame shift mutation is what? When we are just changing the one nucleotide, it will change the whole sequence. Like first they are coding for leucine, alanine, cysteine, but later on, when we just change only single nucleotide, what will happen? They will code for different things. Like here DCA. Now it will code for D, instead of DCA, it will be CGC. So everything will get changed and we will get different amino acids. Deletion is there. Now again the genetic code, which is the dictionary that corresponds to the sequence of nucleotides and the sequence of amino acids. So genetic code is a set of rules by which information encoded in the genetic material. It was given by George Gambo. These are the scientists who understand how proteins are coded began after the structure of DNA was discovered by Watson and Crick. So again, we will use three nucleotide bases. We will use three base pairs to form the combination of 64 codons. These 64 codons, they will code for 20 amino acids. So this is a table. This is a table which codes for 64, which consists of 64 codons. These 64 codons, they are coding 20 amino acids. So genetic code is a dictionary consists of genetic words which are known as codons. Each codon consists of three bases. There are 64 codons, 61 codes for 20 amino acids, and 3 codons are there which are the stop codons UAA, UGA, UGA, UAG. They will not code for any amino acids. So we have start codons, stop codons, sense codons. The anticodon is basically what? These are the sequences of tRNA which pairs with the codon of the mRNA, and during translation this is known as anticodon. So this is during the tRNA. This is during the translation where tRNA have the anticodons of that particular codon sequence. So difference between codon and anticodon. Codon would be present in both DNA and RNA, but anticodon is always present in RNA and never in DNA. So codons are written in 5' to 3' direction, whereas anticodons, they are usually written in 3' to 5' direction. So anticodons of some tRNA molecules have two pairs with more than one codon. This is again the properties of the genetic code. It is triplet in nature, comaless, non-overlapping. The codon dictionary is there, degenerating code, universality, non-ambiguous, initiation code, and terminating code. So we have discussed all these points before also, that it has some polarity. They have degeneracy in case of genetic code. Google hypothesis is there. Yes, this is a very important term. What is Google hypothesis? So It was Crick who postulated the Woburn hypothesis to account for degeneracy of genetic code. Now, what is degeneracy? It means that the first two bases of a codon pair according to the normal base pairing rules with the last two bases of the anticodon. Base pairing at the third codon of a position is Woburn. Sometimes what happens? Only the pairing between first two codons or first two bases is important. The last base pair is not that much important. So the last base pair at the third position is known as Woburn. So Woburn hypothesis explains the degeneracy of the genetic code, that is the existence of multiple codons for a single amino acid. That means for a single amino acid, instead of one codon we have 3 or 4 codons. So there are 61 codons. The number of tRNA is far less, which is due to the wobbling. So if the code were not degenerate, 20 codons would designated amino acid and 44 would lead to chain termination. You have to keep this thing in mind that the codons are degenerative in nature. This is the flow of genetic information from DNA. We will get RNA and RNA to protein. These are the gain mutations. Same points are there. We have initiated codon. Wobbling hypothesis is there that at the third position there is some relaxation. So the rules of base pairing are relaxed at the third position so that a base can pair with more than one complementary base. Some tRNA anticodons have inosine at the third position. Inosine can pair with U, C or A. This means that we do not need 61 different tRNA molecules, only half as many as are required. So if you are able to see, this is proline. This amino acid is coded by these 4 codons. The first two are same. The first two base pairs are same. The only difference is in the last base pair. So the first 3 codons can be recognized by single tRNA having inosine at the first place. This is silent mutation, missense mutation, and some properties of the genetic code. So are you getting it? Is it clear to you? Please let me know. I will share this PPT so that you will able to go through it by your own self also. This is a small topic which is left yesterday when we are doing transcription and translation. So I think let us complete genetic code first. So are you getting it or not? Please let me know. Please let me know, are you getting it or not? So this is the thing which is there in case of genetic code. Now after genetic code we have the next topic which we have to cover. So we have covered all these topics: lack of brain, structure of a gene, ultrastructure of cell, cell organelles, chromosome, stereotype, ideogram, mitosis, DNA, RNA, replication, transcription, genetic code, linkage, then some mutations also we have completed these parts. The only part which is left now, is this 4 and 5. So 4 and 5, which includes methods of induced mutation and CIV technique, multiple factor hypothesis, cytoplasmic inheritance. So we will start with it. We will start with cytoplasmic inheritance. Then we will have to start This cytoplasmic inheritance is also known as extranuclear inheritance. Then after it we have another, which is the multiple factor hypothesis. So we have the multiple factor hypothesis also. Yeah. So we have multiple factor hypothesis, and we have multiple factor hypothesis. Genetic code we have done. Now we are left with cytoplasmic inheritance. Okay. Yeah. So shall we continue with cytoplasmic inheritance? Then we will left with multiple factor hypothesis, or we will start multiple factor hypothesis first. Okay, so we will start with multiple factor hypothesis, also known as polygenic inheritance. So this is known as multiple factor hypothesis or polygenic inheritance. So what is basically multiple factor hypothesis? Now, qualitative traits which are governed by Mendel's law of inheritance, these are clear-cut results. You are able to know that qualitative traits, like we have purple or white color, the clear-cut color is there. Round or wrinkled, the clear-cut color is the clear-cut shape is there. Yellow or green. So these qualitative traits, they are very clear-cut. There is no intermediate phenotype. So either it will be purple or it will be white. Either it will be round or it will be wrinkled. But there is no intermediate phenotype in between there. So variation in qualitative trait is known as discontinuous variation. So the qualitative characters, they are very much distinct. There is no intermediate phenotype in between. But what happened? This is according to Mendel's law, but there are some another phenomena also. So some of the important characters in cultivated plants and domestic animals, as the yield of seeds, fruits, as well as eggs, amount of milk, meat produced by animals, etc., do not fall into two clear-cut classes. So instead of black and white, we will have mixed things. Instead of red and white, we will have mixed things. So these are known as quantitative characters, and the variation is known as continuous variation. So this is your homework also. You have to tell me what is qualitative characters, qualitative traits and quantitative traits, and continuous variation as well as discontinuous variation. So in which we have clear cut distinction. These are known as qualitative characters and the variation is discontinuous. But in which the clear-cut distinction is not present, that is known as quantitative characters with continuous variation. So the quantitative characters, they show continuous variation. Mendel method of analysis is hard to apply in such continuous characters because they seem to mix or blend with each other instead of giving us the segregated in the offspring of the hybrids. So this is the two terms which we have to keep in mind, qualitative character as well as quantitative character. Although we have discussed this also before, the example is skin color in humans, the eye color, the color of the eye, we have discussed it also before. So, the problem of inheritance of quantitative factors was taken by Nielsen Ahle and American scientist East. Okay, these quantitative factors. These investigators showed that there is some blending inheritance. In case of quantitative characters, in case of continuous variation, there is some blending inheritance that can be explained by supposing that Continuously varying characters are due to combined or cumulative action of two genes. So, continuous variation or continuous characters are due to the combined or cumulative action of several genes, each of which exerts a small effect on the same character. Such genes are known as cumulative genes, additive genes or polygenes. So, in this polygenic inheritance, we will have to study cumulative genes, additive genes, or you can say polygenes. So, these cumulative genes is one which, if added to the another identical or similar gene, affects the intensity or degree of expression of a character. So, when the gene is present alone, it has some different effects. When the genes are present together, they are in the dominant form, they are co-dominant, what will happen? The results will be different, or you can say the intensity or degree of expression will be different. So, in other words, a quantitative character is simultaneously governed by several genes. So, many genes are controlling a quantitative character. So, quantitative character is simultaneously governed by several genes, and the effect or action of such gene is known as cumulative or additive in nature. This is also known as multiple factor hypothesis. Now, what is multiple factor hypothesis? When the quantitative trait is controlled by many genes or by polygenes or by cumulative genes, this is known as the multiple factor hypothesis. So, the effect of these genes is known as cumulative or additive in nature. These genes are controlling the quantitative. So, gene pairs which act in a cumulative way to result in a quantitative trait are known as multiple factors. Since quantitative inheritance is controlled by many genes, this is also known as polygenic inheritance. When it is controlled by many genes, it is known as polygenic inheritance, or it is also known as multiple factors which are controlling the quantitative. It is a multiple factor hypothesis in which many genes or polygenes, they are controlling the quantitative inheritance. So, if you are able to see, these are the various shades of wheat. Wheat is in dark red in color, light red, light brown. So, this is only the variation in the wheat only. So, if you are able to see, there is so much variation in the color of wheat. So, various shades of wheat grain. So, these are some examples of polygenic or multiple factor hypothesis. We have grain color in wheat, ear length in maize, ray size in flower heads of composti, seed color in wheat, corolla length in tobacco, skin color in humans, height of man, even your eye color. All these are controlled by many genes. So, a few examples of this polygenic inheritance is described below. We have seed color in wheat. So, this was given that by the scientists who crossed two varieties of wheat, red and white. So, when red and white varieties are crossed, instead of getting red color, which is a dominant color, but we will get intermediate between red and white. That means light red color we will get. Okay, so demonstrating as if red seed color was incompletely dominant over white seed color. That means red color is not dominant over the white color, because in the F1 generation what we will get in the F1 we will get light red color. So when the F1 hybrid plants were self-fertilized, the F2 progeny showed a ratio of 15:1. The red progeny, however, carries various shades between red and white. So we will get so many shades, and the ratio is 15:1 instead of 9:3:3:1. So This is the case of multiple gene hypothesis. So in this example, two pairs of segregating genes are responsible for the color variation in the wheat kernel. The red kernel wheat carries two pairs of dominant genes, both of which contribute same quality of redness to the grain. So if you are able to see, we will get red color, dark red, medium, light, white. So this is the example of polygenes, or you can say polygenic inheritance. Instead of red and white only, we will get red, dark red, medium red, light red and white as well. Similarly, skin color in human, we already discussed it before also, that instead of negro and white only, we will get dark, we will get mulattoes, we will get fair. So all these are the variations in case of skin color. Then corolla length in tobacco, this is also another example. So instead of a the length will be vary. There is not a the length will be from 93 mm to 40 mm. So this is the salient features of quantitative or polygenic inheritance, that it is controlled by multiple genes. Expression of polygenes results in continuous variation. It leads to continuous variation. There is a marked effect of the environment on the expression of genes. Polygenes can be subjected to statistical analysis in order to assess their number regarding a particular trait. So each gene produces a unit effect or individual effect, hence the effect of genes are additive or cumulative in nature. So please read these properties of polygenic inheritance. So polygenes have a pleiotropic effect, that is may influence more than one trait. So this is the difference between qualitative and quantitative inheritance. This is again your homework. You have to tell me the difference between qualitative and quantitative characters. That's all for multiple factor hypothesis. So are you getting it? Everything is fine? Please let me know, are you getting it properly? After it we have another term, another topic which is left, which is cytoplasmic inheritance. So then we have this. After this multiple factor hypothesis, we have another term which is cytoplasmic inheritance. So is it clear to all of you? Please let me know, then I will start cytoplasmic inheritance also. So shall we continue with cytoplasmic inheritance now? Please let me know. So shall we continue with now cytoplasmic inheritance? Please reply. Are you able to listen to me? Please reply. So now we have only one topic, which is the cytoplasmic inheritance, which is there. So now I am just going to share the PPT of cytoplasmic inheritance. So now we have another term which is here. We have cytoplasmic inheritance. Are you able to listen to my voice? Please, any one of you, I am audible. Please reply to me. I am audible to you or not? I'm audible? Okay, okay, thank you, thank you. Now we have on only one topic which is there, which is cytoplasmic inheritance. I am going to share the PPT. Yes. So now we have cytoplasmic inheritance. It consists of maternal inheritance. We will have to study mitochondrial or maternal inheritance. We will study chloroplast inheritance, then inheritance involving infective particles, that means kappa particles in paramecium. We will study that also, and at the end there will be a significance only. Now there is some inheritance which is there because of the genes which are located in the cytoplasm. So the plasma genes are located in the DNA present in the mitochondria and in the chloroplast. Together both the DNA are called organelle DNA. Therefore, this type of inheritance is often referred to as organellary inheritance, plasmid inheritance, or also known as mitochondrial inheritance. So the cytoplasmic inheritance which is occurring in the cytoplasm, it is of three types. We have maternal inheritance, organellar inheritance, and inheritance involving infectious particles. So in this, generally the character of only one of the two parents is transmitted to the progeny. Basically the characters from the mother, from the female, it is transmitted to the progeny. So hence such inheritance is usually referred to as extranuclear or extrachromosomal or maternal or uniparental inheritance. So this inheritance is usually referred to as extranuclear or extrachromosomal because it is not occurring in the nucleus, it is not occurring inside the chromosome, rather it is occurring outside the nucleus in the cytoplasm. So it is known as nuclear extra chromosomal maternal inheritance because only the character from the mother will be passed to the next generation. So in most animals, paternal mitochondria enter the oocyte cytoplasm after the fertilization, and then their DNA is never transmitted to the offspring because in the mitochondrial DNA only the character from the mother will be passed to the next generation. So this type of mitochondrial DNA inheritance is known as maternal inheritance because the characters from the father, the characters from the paternal will not be there in the upcoming generation. So this is the examples of these. The number one is shell coiling in snail. So this is a snail. In snails, the shell is spirally coiled. Snail exhibits two types of coiling. So the shell is coiled to the right, it is dextral. When the shell is coiled to the left, it is known as sinistral. So we have two types of coiling of snail's shell. It will be either dextral or sinistral. So the gene for dextral coiling is dominant D, and for sinistral coiling it is recessive small d. So Boycott and Driver showed that the character of a coiling is determined by the gene of mother and not by the individual's own gene. So this coiling or this character of coiling is determined by only by the mother's character, not by the individual's whole gene. So there is a crossing of dextral with the sinistral. Dextral is dominant over sinistral. After crossing, what we will get? We will get dextral in the ratio of 3 and sinistral will definitely 1. But this dextral ratio is concerned with the maternal character only. So the coiling depends on the genotype of egg donor parent regardless of the phenotype of that parent. If the parent is DD or DD, the offspring will be dextral. So this is the case of maternal inheritance basically. Then another inheritance which we have, which is known as cytoplasmic inheritance. Now what is cytoplasmic inheritance? The true cytoplasmic inheritance is one which involves plastids or chloroplasts and mitochondria. The cytoplasmic inheritance is again of two types. We have two types of cytoplasmic inheritance, like we have mitochondrial inheritance which occur in the mitochondria and plastid inheritance which occurring in the plastid. So the cytoplasmic inheritance is governed by the genes which are found in the chloroplast and the mitochondria. The genes which govern a cytoplasmic inheritance are C are called plasma genes or cytoplasmic genes. So this is an example in case of Mirabilis jalapa. The cytoplasmic inheritance was found by Carl Cons in Mirabilis jalapa, also known as 4 o'clock plant. He studied inheritance of leaf variegation in Mirabilis jalapa. What he found? Variegation refers to the presence of white, yellow spots on the sides on the green background. So on the green leaf we have white or yellow spots. So in Mirabilis jalapa, leaves may be green, white or variegated. Some branches may be green, only white, only variegated. So Coran made crosses in all possible combinations and produced these three types of branches. So he found the observations are flowers on wholly green branches produce seeds that grow into normal plants. So those flowers which are there on the green branches, they will produce normal plants. Flower on variegated branches yield offspring to three kinds of green, white and variegated leaves. Flowers on the branches wholly white, they will produce seeds that grow into white plants without chlorophyll. So we have three types of color. We have green leaves, variegated and white leaves. So green leaves will give normal plants, white will give white plants without chlorophyll. Then we have cytoplasmic sterility also. The inheritance of some character is governed by mitochondrial DNA. The examples of mitochondrial inheritance include cytoplasmic male sterility in plants, in neurospora and even in yeast. Then after it we have another term which is kappa particles in paramecium. Now what are kappa particles? It was born in 1943, described the inheritance of some cytoplasmic particles known as kappa and their relation to the nuclear gene in paramecium. There are two strains of paramecium, one strain is killer and the other is sensitive. The killer secretes a toxic substance called paramycin which kills the sensitive. So the paramecia of the killer strain contain large number of kappa particles in their cytoplasm. The production of kappa particles is dependent on a dominant allele K, so that the killer strain are K or capital K small k and sensitive strains are only small k. So this is the kappa particles which we have. So this is some significance of this cytoplasmic inheritance which is occurring in the cytoplasm rather than in the nucleus. So what will happen? It is useful in explaining the role of various cytoplasmic organelles in the transmission of character in different organisms. So studies of cytoplasmic inheritance have played key role in mapping chloroplast and mitochondrial genome in several species. The CMS cytoplasm can easily transfer to various agronomic bases for their use in the development of superior hybrids. The role of mitochondria in the manifestation of heterosis is gaining increasing importance these days. So mutation of chloroplast DNA and mitochondrial DNA leads to the generation of new variants. Some of such variants are of special significance, especially in the ornamental plants. So according to the Mendel, the ratio is 3:1 and in F2 the ratio is 9:3:3:1. But this Mendelian inheritance was found only in the nuclei. But some inheritance is also there which is not in the nucleus, that is known as extranuclear inheritance or cytoplasmic inheritance. That inheritance which is not in the nucleus, rather it is taking outside in the cytoplasm, this is known as cytoplasmic inheritance. Or you can say extra chromosomal or maternal inheritance. So the genes governing the character showing non-Mendelian inheritance are located outside of the nucleus and found in the cytoplasm. These genes are known as plasma genes or cytoplasmic genes or cytogenes, or also extranuclear or extrachromosomal genes. So these are the Mendelian inheritance. This is non-Mendelian inheritance. In Mendel, we have nuclear genes. In case of this, we have plasma genes, or you can say cytoplasmic genes. The examples are these one. Plastid inheritance in Mirabilis, shell coiling in snail, kappa particles in paramecium, cytoplasmic maize sterility in maize, milk factor in mice. All these are the examples of non-Mendelian inheritance. So we have maternal effect, in which only the character from the mother will pass to the next generation. Inheritance involving infected particles, cytoplasmic inheritance. So this is maternal inheritance. The example is coiling pattern of shell in snail. We already discussed it that in the snail we have two types of, all the strains will be killer type only. After it we have cytoplasmic inheritance in case of Mirabilis jalapa, in which we have three types of leaves: green leaves, white leaves and mosaic leaves. So the green branches contain green plastids in their leaf. The mosaic branches contain green and white plastids, and white branches contains only white plastids. So if you are able to see, when we are crossing the green leaves with white one, in the F1 generation we will get white only. Now when we are crossing green leaves with white one, we are getting green. So it only depends on the character by the mother. So if the mother is white, the offspring will definitely white. If the mother or the maternal inheritance is green, then the offspring will be green only. So the character is here only because of the character which is there present in the mother. So if father is white or green, it doesn't matter. The character given by the maternal only will be passed to the next generation. So this is the example in case of Mirabilis jalapa. This is the mitochondrial inheritance. This is the inheritance in which the character from the mother will be passed to the next generation. So this is the maternal effect inheritance. So this is Carl Cornins in 1909 who was able to found different colors in Mirabilis jalapa, or you can say the leaves are also of different colors, but it is because of the maternal inheritance. Similarly here, if the mother is sinistrial, then definitely in the next generation everything will be sinistrial. If the mother or if the character from the maternal is dextral in nature, then in the next generation it will be dextral only. So this is the you can say snail. This is the coiling of shell in case of snail. So we will have the dextral if the parent, if the mother or if the character by the maternal inheritance is dextral, then it will be dextral all. This is Sean born in 1938 who was able to let us know about the killer strain. So this is the killer strain, and it consists of kappa particles. These kappa particles will then move into the sensitive strain and change the sensitive strain into the killer strain again. So these characters, they are passed from the killer strain to the sensitive strain, and now they both become the killer strain. So that is the whole thing which are there in case of cytoplasmic inheritance. I hope you get it and you are getting something. I hope so. So is it clear to all of you? Now we have the last topic of this generic session is left, which is the CIB technique. Okay, so the last thing which left is the CIB technique. We have done multiple factor hypothesis, cytoplasmic inheritance we have done, in which instead of nuclear inheritance only some characters are there which will be transmitted by the maternal inheritance. Now the last thing which is left is the CIB technique. So let me open the CIB technique now. After it you are syllabus will be completed. Okay. So now we have the CIB technique. We have methods of inducing mutations, as well as we have the CIB technique. I'm now just going to complete it because then I will share the PPTs and PDFs with all of you. Okay. So the last is the CIB technique. Now I'm just going to complete the topic with the CIB technique. So are you getting it? My voice is audible to all of you. Please let me know. My voice is audible. Now we will move further with the CIB technique. So there are methods of inducing mutation and CIB technique. So mutations can be induced by a number of agents. Like what are basically induced mutations, which are basically by some chemicals. The agents are capable of inducing mutations are called mutagens. Mutagen is a natural or human-made agent which can alter the structure or sequence of DNA. The different mutagenic agents may be classified into following groups. We have physical mutagens as well as we have the chemical mutagens also. Are you getting my voice? Are you getting my voice? So I am just letting you know about the mutagens which are here. We have physical mutagens as well as we have the chemical mutagens which are there. So physical mutagens are basically what? Yeah. So physical mutagens are the different types of radiations having mutagenic properties are known as physical mutagens. Like we have UV rays, X rays, gamma rays, cosmic rays. They all come under the physical mutagens. Radiations are there. So radiations were the first mutagenic agent known. Its effects on the genes were first reported in 1920s. So these are the radiations. So radiation itself was discovered in 1890s. So radiations are grouped into two classes depending on the effect of they have. We have ionizing radiations, we have non-ionizing radiations. So these radiations they comes under the physical mutagens. Then after it we have some chemical mutagens also. We have already covered it, but I am now because I just start the PDF, so I let you know. So now what are the chemical mutagens? Chemical mutagens are the chemicals like we have bromouracil, we have aminopurine. So all these chemicals they are able to induce the mutations. They are able to they are able to make the change in the gene or variations in the gene. So these chemicals structurally resemble purines and pyrimidines and may incorporate it into the DNA in place of normal bases. So during DNA replication, instead of purines and pyrimidines, these bromouracil and aminopurine they will bind in place of normal bases. So these adenine analog too can pair with thymine and cause AT to GC or GC to AT transition. So these make the changes in the gene sequence. So these are some another chemicals. We have nitrous acid, we have gonadine, methyl methane sulfonate, ethyl methane sulfonate. They are also the chemicals which can make the changes in the gene. Some intercalating agents are also present like acridine orange, proflavin, ethidium bromide. They are flat multiple ring molecules which interact with the bases of DNA and insert between them. This insertion causes the stretching of DNA double helix and the DNA polymerase is fooled into inserting an extra base opposite an intercalated molecule. The result is intercalating agents cause frame shift. So because of these intercalating agents, frame shift mutation is there. Because of these intercalating agents, frame shift mutation is there. So these are the mutations which are induced by humans. These are those types of mutations which are there because of some chemicals, because of some reagents. So agents altering DNA structure, this is used as a catch-all category which includes a variety of different kinds of agents. These may be, now we have some large molecules which bind to the bases in a DNA and cause them to be non-coding. Agents causing intra or interstrand crosslinks, chemicals causing DNA strand break. So all these are the mutations which are there, which are induced by the humans. Now how to detect the mutations? So these are the mutations will be detected by the phenotypic expression, by the phenotypic appearance, like we have Turner syndrome, we have Klinefelter syndrome. All these mutations can be detected by the phenotypic expression only. So this is the detection of the mutation. Now we have a technique which is CIB technique. Also this is there in the previous slides also, which we have covered yesterday. But what is CIB technique? So this method was invented by Muller and used for the unequivocal demonstration of mutagenic action of X-rays. So Muller was the one who was able to know about the action of X-rays, about the changes which was bring by X-rays only. So in this method, females containing one normal X chromosome and another X chromosome which contains CIB containing extra 3 genes are used for the analysis. Out of the 3 extra genes, one gene suppresses crossover, the other is reversibly lethal in heterozygous condition, and the last gene is semi-dominant marker which is known as bar B gene. So females containing CIB chromosome are called CIB stalk Drosophila. So this was done, this type of mutation was by X-rays was noted in Drosophila. The normal males are exposed to mutagenic sources for a fixed period and then mated to the CIB stalk Drosophila. Males containing CIB chromosome will die due to the effect of lethal genes, whereas normal males and females, both normal and with CIB, will survive. So females with CIB chromosomes and identified by Baird phenotype are selected and crossed with the normal males. In the next generation, 50% of the males, they will get this CIB gene and they will die. So if the mutation has occurred in the normal X chromosome, then even the normal male will die. So if no mutation has occurred, all the other 50% of the males will survive. The frequency of lethal mutation can be accurately scored in large samples. This technique is simple, rapid, and there is little chance of any error. So I just want to show you a diagram. Maybe in this we have a diagram. So again, this is the mutation, the types of mutation, the nature of mutation. I will share this PPT, you can PDF. This is substitution, we have insertion, deletion, frame shift. Kindly read it by your own self. So I just want to tell you about the CIB technique, which is for detecting the mutations by the X-rays. Yeah. So this is the CIB technique. A cross is made between CIB female. So CIB method was developed by Muller for the detection of induced mutation. Though this method was invented by Muller and used for unequal demonstration of mutagenic action of X-rays. So females containing one normal X chrome and another X chrome which consists of CIB consist of 3 X genes for the analysis. So if we are crossing the CIB female with the normal male, what we will get? We will get CIB female as well as we will get CIB female as well as we will get CIB male. But the CIB male will die, only the CIB female will survive. So now in the second cross, we are crossing again this CIB female with the normal male. Okay, so this is a normal male mutagen treated. Now this is a CIB female with normal male only. Now what will happen? The normal male will the normal male dies if lethal mutation is induced. Now this CIB male will dies also. So now a cross is made between the CIB female and normal male. This time the CIB female has one CIB chromosome and one mutagen treated chromosome received from the male in the earlier crosses. So what is the what basically he wants to tell us? This produce two types of female, half with CIB chromosome, half with mutagen treated chromosome. Both the progeny will survive. In case of males, half with CIB will die and other half or have mutagen treated chromosome. So if a lethal mutation was induced in mutagen treated X chromosome, the remaining half males will also die, resulting in the absence of male progeny. So the male progeny will get absent. Absence of male progeny confirms the induction of sex-linked recessive lethal mutation in the mutagen treated with Drosophila male. So this is the thing which is there in the CIB. You have to remember only one point. You have to keep in mind in only one point that this technique is basically used by Muller in case of Drosophila. Why he used this technique? Because to know the mutations which are there because of the X-rays. Because of the X-rays, the mutations which are there, the female, if the female is able to survive with CIB, but the males will die immediately. So this is the CIB technique which was there by Muller. This is only the mutation which is given in the Drosophila by the help of X-rays. So I think that's all for our today's lecture series which we have. We have to cover the important topics. We have covered mutations, we have covered multiple factor hypothesis, cytoplasmic inheritance as well as genetic code. And all these topics are quite important. I will ask you, I will suggest you to read these topics in detail. Now the homework which you have to do, you have to, what is the first week homework which I told you? I gave you some, yes, I gave you quantitative and qualitative characters. Along with it, you have to tell me the properties of the genetic code. Please, any one of you, please write it somewhere. The properties of the genetic code. Number one, the properties of the genetic code. Then quantitative and qualitative characters along with the example and the difference between those. And last, you have to tell me the examples which are there in the cytoplasmic inheritance. The examples in case of extra chromosomal cytoplasmic or maternal inheritance. And the last thing you have to tell me about this CIB technique. So is it clear to all of you? Can you do it? Please, any one of you, please write this homework in the group so that all of you will be able to do it. Is it clear to all of you? Please let me know. So I hope everything is clear to all of you. If you have anything to ask, you can ask me. Today is our last lecture series. From after it, we will not meet on this YouTube live stream. So if you have any question or query, you can ask me. Otherwise, I will reply you only by the mails. I will reply you on the Telegram also, but I will have to check your abstracts and you have to focus on the review paper. So if the question is clear to all of you, please reply. Any one of you, please reply. If you have any question, please reply. If the homework is clear to all of you, please let me know. Any one of you, please. So, yes, So Atria, please try to write the homework in Telegram also, so that all of you will able to send me your assignments as soon as possible. So, if the homework is clear to all of you, you have to let me know about quantitative and qualitative traits, the difference which are there, the properties of the genetic code, the extra chromosomal inheritance, example of extra chromosomal inheritance, and in the last, the CIP technique. So, is it clear to all of you? Please let me know. Today is our last lecture series. If you have any question, you can ask me. If you have any question or something, you can ask me. Otherwise, this is our last lecture, so we will not meet from tomorrow on Telegram on this YouTube live. So that's all from my side. I wish you all the best for your future, and be good in everywhere. Okay, so stay good, work hard, and always shine in your future perspectives. That are the best wishes from my side for all of you, and take care of yourself. So from tomorrow onwards we will not meet on YouTube live or on any other platform. So that is all for this lecture series which we have together. Now the only thing which is left is the review paper on which we have to work together. So that's all from my side. Take care of yourself, and you have a great years which will be coming now. So wish you good luck for your future perspectives. Take care of yourself and stay happy, stay safe. So that's all from my side. Take care of yourself, and anyone please write down the homework in the Telegram also. So that's all from my side. Bye bye. Take care. Bye bye to all.